I am going to prove to you why pyruvate dehydrogenase is the answer to clue 5 by going through the clue one by one. So, pyruvate dehydrogenase inhibition means that there's no conversion of pyruvate into acetyl-CoA, means that the Krebs cycle is essentially inhibited. No citric acid produced, no aketoglutarate produced, means no FADH2 and no NADH produced at the end. But it allows fatty acids to be made from glucose. Now, lipogenesis must require acetyl-CoA, and because of the inhibition of PDH, there's another more complicated system mechanism that occurs whereby buildup of pyruvate will stimulate pyruvate carboxylase to convert pyruvate into oxaloacetate. Now, oxaloacetate combines with excess acetyl-CoA from leftover to form citrate. And citrate, I believe, is moved to the mitochondria for fatty acid synthesis. But before this, in the cytosol, the citrate is essentially cleaved by ATP citrate lyase to regenerate acetyl-CoA and oxaloacetate. Now, that acetyl-CoA will be used in the lipogenesis to form malonyl, I believe is malonyl CoA, and then there's a couple of steps like reduction, hydration, fatty acid synthase involvement to get to fatty acyl-CoA and your triglycerides. It prevents the synthesis of glutamate from acetyl-CoA, but allows glutamate to enter the Krebs cycle. Then there's two ways glutamate can enter the Krebs cycle without using acetyl-CoA, and the first one is essentially transamination, where you're converting L-glutamate and oxaloacetate from this diagram with the help of aspartate aminotransferase into aketoglutarate and L-aspartate. Now, aketoglutarate is the precursor to glutamate, but there's also a different method, and it is essentially glutamate conversion into aketoglutarate by glutamate dehydrogenase, and that occurs when there's a buildup of pyruvate. That's the stimulus, and that bypasses the need essentially for pyruvate dehydrogenase if it is inhibited. And because aketoglutarate is now produced, that will continue to the Krebs cycle all the way around to produce FADH2 and NADH. Enzyme is inhibited by depletion of NADH pool. That's fairly simple. So pyruvate requires the donation, I believe, of electrons to NAD+, to form acetyl-CoA. Without NAD+ concentration, a high enough concentration, there'll be no acetyl-CoA produced. So the enzyme is also inhibited. Mutations in the target enzymes found in cancer. This occurs due to the Warburg effect. So because there's a mutation of PDH, which is often due to an overexpression of pyruvate dehydrogenase kinase, it reduces the PDH activity and it increases the reliance on glycolysis, as there is less reliance on oxidative phosphorylation because the Krebs cycle is essentially being inhibited. And cancer cells prefer this type of environment, a very simple energy pathway, and that will lead to more anaerobic respiration, which produce more lactate, which will promote tumor growth from metabolic acidosis.